first of these short stories concerns a company called InterX, uh, which has developed a new and powerful approach to computational chemistry for the discovery of drugs by the use of calculation rather than by chemical synthesis and experimentation. The essence of InterX technology is an approach that we refer to as a quantum mechanical polarizable force field, or in brief, QMPFF. I'll explain in a few moments what that entails. I should say that this is an innovation relating to small molecule drug development. It is pertinent to very many applications in the pharmaceutical industry, including what you will hear about after I speak from Asher Nathan about a company in which we are both engaged named Neotex. As many of you are aware, the purpose of small molecule drug development is in very many cases to interfere with ligand protein interaction, symbolized here by a famous sculpture, ligand protein interaction in which the ligand on the left binds in a cleft of the target on the right. The target is often an enzyme whose activity is, for one reason or another, inhibited for the purpose of of pharmacy, of disease therapy, or what have you. The current approach, indeed the most effective one that is employed throughout the pharmaceutical industry for development of small molecules for this purpose of binding in a cleft of a target to interfere with ligand protein interaction, begins with structure determination and is essentially structure-based for the purpose of drug design. Structure in the past always determined by X-ray crystallography, today sometimes by cryo-electron microscopy. Based on the structure, a potential inhibitor of an enzyme or what have you may be designed and then tested by synthesis, by experimentation, to judge whether anything approaching a successful application has been achieved. The process will, in the best case, lead to a molecule with some degree of efficacy, which is then improved by repeating the entire process of structure determination, in this case, a co-crystal structure of the starting molecule with the target, redesign based on the information from the co-crystal structure, synthesis of the next best compound, and again, measurement of the effect upon enzyme or other relevant activity. It's a cycle that is repeated over and over until, in a favorable circumstance, or the best case, a molecule with the desired efficacy, a molecule with the desired affinity and impact upon activity of the target is achieved. Typically in the industry, over the years, this has required, it has occupied the efforts of hundreds of chemists over a period of years. It has involved the synthesis of thousands of molecules, arriving in eventually hit or miss fashion at a possible drug and a drug candidate for the desired purpose. This occasionally succeeds. More often than not, it fails, but it is the best approach available at the present time. Now, it has been hoped and indeed tried, attempted for very many years to facilitate the process, to speed it up, and to achieve a better result by computation of small molecule protein target interaction. This entails, of course, knowledge of the force involved in the interaction. What is required for the computation are force fields. And in every such undertaking to this point, except for the one I will describe. The force fields are classical. This is true in every academic study. It is true in every industrial application. It is true of the companies that deliver uh, products for this purpose, as well as the pharmaceutical companies that utilize them. The force fields are classical in as much as electrons, which are responsible for the interactions, are represented as point charges, as individual negative charges. Uh, a classical force field cannot represent any other interaction except an electrostatic interaction. In particular, it cannot represent nonpolar interactions, what some of you will know as van der Waals interactions, equally often more important in small molecule protein interaction. Uh, the interactions of this second category depend on polarization, which is impossible to calculate on the basis of a classical force field. This slide illustrates the failure of classical force fields. It shows they simply don't work. They're easy to do, they're rapid to calculate, but they simply don't work. What you see in the lower right is a plot of the energy of interaction measured experimentally, plotted against on the horizontal axis, the calculated energy of interaction. These circles are the results of the calculations, and what you will see is that they bear no resemblance to the actual experimentally measured energies. There is essentially a random relationship between the hoped for calculation of the result and that actually found. The alternative, which has been developed at InterX that I mentioned before, treats electrons not as point charges, but as clouds. We know from quantum mechanics, which provides a proper and an accurate description of chemistry, that electrons are indeed only appropriately represented in this manner. When electrons are represented as clouds, then polarization is possible. A polarizable force field can finally describe polar interactions such as van der Waals interactions. The quantum mechanical polarizable force field includes an electrostatic component, illustrated here, as well as the interactions formed by bonds between atoms, also a component of a classical force field, but additional terms in the energy which are purely quantum mechanical in origin, such as electron exchange, what is called dispersion interaction, the energy of induction interaction. The image on the lower right illustrates electron clouds and their polarization, and it shows that process of polarization as it occurs in a dynamic manner, which can be calculated, and here was actually derived from the computation using the quantum mechanical polarizable force field. What you see on the lower right is the actual electronic structure of a water molecule, an oxygen with two hydrogen atoms, which interact in a manner which includes both electrostatics and quantum mechanical features such as polarization. A quantum mechanical polarizable force field is complicated, far more than the current classical force fields in use in all other work, which are straightforward, simple to calculate from a straightforward algebraic expression. The problem of a quantum mechanical calculation is illustrated here. So a full quantum mechanical calculation, which represents a solution to the Schrodinger or the wave equation that precisely describes all of chemistry, is hopelessly difficult and time consuming. It would take at least a million billion years with current computational capabilities. It would yield a highly precise result. A classical force field, such as illustrated on the by the symbol on the lower right, a classical force field, in the simplest case, simple docking of molecules to their targets computationally, is extremely rapid, but gives a result that is hopelessly imprecise and cannot be used to guide a drug development procedure in a useful manner. Finally, uh, this is stuck now, sorry. Uh, oh, there we go. The conventional force fields, the classical ones that I mentioned, represent a slight improvement in both respects, but still far from the necessary accuracy for guiding a drug development process. Finally, the quantum mechanical polarizable force field developed by InterX and uniquely uh, possible through the approach devised by that organization supplies both the necessary accuracy and a speed that is within the realm of possibility. At present, a calculation uh, with very high accuracy takes several days. In the best version of the software now available, a matter of hours, and in time, we believe only minutes or seconds. Here you see the results of application of the quantum mechanical polarizable force field in the gold-colored circles on the plot. So the results of calculation are plotted on the vertical axis, the experimental measurements for interaction of a molecule with the enzyme lysozyme on the horizontal axis, and you see that the orange dots fall beautifully on a straight line, indicating a near perfect correspondence of calculation with experimental measurement. The pink triangles and the purple squares represent results of the best available classical force fields that are widely used in the industry today. And you see the results are really, by those approaches, not better than random. And so the computational efforts that are possible today in the industry outside of what I've described, developed by InterX, are really worthless and cannot be used to replace chemistry. As an example, as a proof of concept, we recently explored the application of the InterX approach, the quantum mechanical polarizable force field, to poly-ADP ribose polymerase inhibitors. PARP inhibitors are, as some of you will know, already approved for monotherapy for BRCA mutations, and they're presently employed in a very large number of combination trials with other chemotherapeutic agents. In our application of InterX technology, we actually started from scratch. We devised 3 new starting scaffolds. By computation, we could explore 60,000 possible compounds over the course of a few weeks. We had 26 molecules synthesized, only 26, not thousands as conventionally done, with the chemical facilities available to us. That took a period of several months. We then measured assays both in vitro and in vivo. We could show that the molecules that we derived had 100-fold higher affinity than any of the PARP inhibitors currently in the clinic. The currently approved inhibitors have affinities in the range of nanomolar. The molecules that we discovered rapidly and cheaply in this manner by exploring such a very large number of possibilities have affinities in the range of 10 picomolar.